Sure we're all very familiar with the picture before us, the three-domain model of life, with bacteria, archaea, and eukarya all split between their last common ancestors. But how many of us are familiar with this new two-domain model of life, where we have eukaryotes coming from the archaea rather than them being a separate domain of life? Well, the three-domain model of life was first posed by Carl Woese in his 1990 paper Towards a Natural System of Organisms, a proposal for the domains archaea, bacteria, and eukarya. This was based on his research into the rRNA sequences of different organisms. But first, a quick lesson on taxonomy. This has been going on for thousands of years, and even Aristotle wrote in his Lost Works of the Creatures of the Sea, as well as in his Historia Animalia, about the tree of life and how he thought the plants and animals on Lesbos were interrelated. Since Aristotle, there have been many famous taxonomists, number one, of course, being Carl Linnaeus, the father of modern taxonomy himself. He published in the Systema Naturae all the names of the plants and animals in the world that he knew of and how they were related to each other. However, Linnaeus did not know what to do with van Leeuwenhoek's discovery of bacteria. Originally, he actually put them under worms. Van Leeuwenhoek was a Dutch lens maker, and looking down his lenses he saw all sorts of things that we did not know existed, such as nerves and capillaries and the shape of sand grains. And of course, bacteria. And that was maybe where we were in the early part of the 19th century, where we had plants and animals known, which we knew existed, and then we had all sorts of other microorganisms, like down here in the bottom of this phylogenetic tree we can see bacteria and algae and fungi, and no one was really sure where these fit. Were they plants or were they animals? Well, obviously they were plants, because they didn't move and they weren't active and they weren't made out of flesh like animals do, because at the time ideas in science would have been pushed forward based on ancient philosophical principles, which, since we stem from the Greek tradition, would be the animal spirit. But not everyone was pro the two-domain model of life. Haeckel, for example, proposed a three-domain phylogenetic tree with plants, protists, and animals, protists in this case being any single cellular organism. And this makes a little bit more sense. Haeckel realized that plants and animals are completely separate things, because of course they are, but these microscopic versions of life must be related somehow to plants and animals, and he put them in between plants and animals, because plants are passive and animals are active, and you can see things move around, so therefore they must be sort of in between both. In the middle of the 20th century, however, this is sort of where we were looking at. This is Whittaker's five-kingdom system based on three levels of organization. The prokaryotic kingdom, which is Monera, the eukaryotic unicellular kingdom, which is now called Protista. Then on top of that you have Plantae, Fungi, and Animalia. These he separated out by their main modes of nutrition: photosynthetic, absorptive, and ingestative. Now, this model was one of the predominant models that were existing at the start of our story, when Carl Woese decides to find out how exactly bacteria are related to each other and whether or not we can properly classify them. At the time, a lot of work was going into phylogeny, trying to work out how bacteria related to each other and the other domains of life that we knew at the time. Sanger had managed to create a good sequencing technique by 1965, but this was still not popular because of its expense and time-consuming nature. Zuckland and Pauling instead wanted to use amino acid substitutions in highly conserved proteins in order to track phylogenetic drift. Though since not all proteins are conserved across all domains of life, and the fact that these proteins might not retain the same function, this was not the way Woese wanted to look at things. Woese wanted to look at rRNA. In this excellent review, Bacterial Evolution, in 1987, 10 years after he discovers the existence of archaea, Woese really goes into and describes why rRNA is the ultimate molecular chronometer, and I highly recommend everyone to read this, just because the manner in which he writes is very interesting, very different to the way people write now, and I would say this is a very interesting look on taxonomy by someone who is arguably one of the greatest taxonomists of all time. In it, Woese explains why rRNAs are the best molecular chronometers. In short, they show a high degree of functional constancy, which assures relative good clockwise behavior. They occur in all organisms, and different positions in their sequence mutate at different rates, meaning that you can measure both very large phylogenetic distances and very small phylogenetic distances. They are large enough to consist of many domains. There are 50 helical domains in the 16S rRNA secondary structure, but also at the time, Sanger's rRNA sequencing method was very good, meaning that you could reliably sequence rRNA of cells. And very humorously, at the end of his point, Woese talks about how in the future a lab could sequence hundreds of rRNAs per year, whereas now, obviously, we do shotgun metagenomics where we sequence thousands of rRNAs at once. It's incredible how fast science has moved forward. So what? Woese does his rRNA experiment, and what does he find? He finds this graph here, which is an association coefficient between the representative members of the three primary kingdoms, as he called them at the time. What he did was digest the rRNA from these organisms, and he put them into a electrophoresis machine to separate out the oligonucleotide fingerprints, and then he pieced them back together. And this is a measure of how similar these organisms were to other organisms. So on the top, you have your eukaryotes. In the middle section, you have bacteria. And in the bottom section, you have methanogenic bacteria, as they were known at the time, or, as Woese tries to call them, archaea. Now, what you can see very nicely here, and you can pause it and look at this diagram, is that there are groupings of numbers that are all very similar to each other. So that top left corner, all the eukaryotes have a very high score close to each other, about 0.3. In the middle section, all the bacteria are quite similar to each other, about 0.2, 0.3. Sorry, about 0.22, about 0.23. But in the bottom right, you have these archaea who are very similar to each other. You have very high scores, 0.25 on average. But if you compare those scores to their scores with the bacteria, you can see they are very distantly related from bacteria. Only 0.01 association coefficient. And across one more, you'll see that the eukaryotes are very distantly related to them also. These are neither bacteria nor eukaryotes. And using this, he decides to put forth his idea for a third primary kingdom. The more oligos I sequenced, the less prokaryotic it felt. Woese knew almost immediately that something was going on here. He was skeptical, and he shared his out-of-biology experience with George Fox, his long-time PhD student at this time, and who would come on to be one of the other forefathers of archaea. They both agreed this was something new. This wasn't just a bacterium, this was something novel. He ends this with, little did we know how much trouble there would be trying to convince other biologists of that idea, and boy was he right. In 1977, these two papers also came out. The first by Fox and Woese, characterizing methanogenic bacteria by their 16S rRNA sequence, which was what led them on to their later work, but also this paper by Kandler, who we'll see again later on, who realizes that peptidoglycan does not exist in the cell walls of these methanogenic bacteria. In a later episode, we'll talk more specifically about the cell walls of archaea, but they are very unique and very cool, especially, like me, you're a bacteriologist. In 1983, Woese publishes another paper looking at the detailed analysis for higher order structure in 16S-like rRNA sequences across a wide variety of life. This is quite possibly my favorite paper ever. If we have a look, it is just a breakdown of dozens of tables looking at every single helix in the rRNA sequence and explaining the differences between bacteria, eukaryotes, and archaea. There are quite literally dozens of tables exactly like this, looking at E. coli, H. vulcani, X. levaeus, yeast mitochondria, human mitochondria, chloroplast, paramycium. Every single angle of looking at this, Woese decided to look at. He was fed up of people talking down on archaea. And just for your viewing pleasure, this is X. levaeus, the African clawed frog, and he's just here because I think he looks amazing. But enough about him. This is what the paper is about, the secondary structure of rRNAs. On the top left, we have the representative eubacterial rRNA. E. coli in this case. On the top right, we have H. volcani. On the bottom left, we have yeast, and on the bottom right, we have the minimal subunit rRNA. Now, there are several key differences here between all of these things. And this is best illustrated from this diagram from Fry's paper earlier on this year describing the history of the discovery of archaea. He highlights the key areas in the secondary structures, so it makes it much easier for us to see the differences. And if you go back to the previous image, you will be able to start to see some of these and some more differences just by scanning and slowly looking through the diagram. Alright, now let's talk about phylogeny. Ever since Woese first described archaea in 1977, the question has been, how do they fit into the tree of life? In 1990, when he described the existence of the domain, he came up with one idea, the three-domain model, which we are all very familiar with. Since then, many different phyla of archaea have been discovered. Some have been reclassified, others have been dissolved altogether. And that takes us to the modern day, where there are now four main groupings of archaea: Euryarchaeota, the Deephan superphylum, the Tax superphylum, and Asgard archaea. Now, what I'm going to do now is go through each of these groups and highlight some of the cool key species to try and pique your interest. First up, Euryarchaeota. And what you'll notice is that a lot of these groups beneath the phylum level are methanogens: methanopyri, methanomicrobia, methanococci. All metabolizing methanogens are archaea. Also in this group are halobacteria and thermococci. The first species is a methanogen, Methanococcus janishi. This was the first archaea and the third ever species to have its whole genome sequenced. You can see really how hard Woese is pushing the idea of a third domain of life, getting its sequence done so fast when sequencing was so hard and so time consuming and so expensive really speaks volumes. It's really interesting to me to see how the whole genome sequence map looks, and I really recommend anyone who does a lot of sequencing, I do microbiome work so we deal with a hell of a lot of sequence data, just to go through this paper and see exactly how they make it look. It's really, really interesting to me, and they look at all sorts of things that I was shocked that they had the ability to do. This species lives in undersea white stack chimneys, and they live about 3,000 meters down, where there's about 295 atmospheres of pressure. These images on the right are taken from a paper where they actually lowered the pressure in the fermenting vessel and they burst. It's a really, really cool paper, and it shows images of these cocci burst. Next up is another methanogen, Methanobrevibacter smithii, which is the main human relevant archaea since it resides in the gut of humans, but also other animals such as horses, rabbits, cows. Usually ruminants is where the methanogen production of ruminants comes from. It has been found in ancient DNA studies, which puts its presence in the human microbiome to as far back as 14,000 years ago, and Kostich's Nature 2021 paper compares two different M. smithii genomes and shows through genetic drift that the two strains have been separated by between 51 and 128,000 years. M. smithii is the only sort of pathogenic archaea because it's been associated with constipation predominant IBS, small intestine bacterial overgrowth and obesity, partly down to its production of methane, slowing down intestinal transit time. However, no direct interactions between this archaea and our own cells has been shown, implying that this is mostly microbiome-based effects. Moving on from methanogenic bacteria, we have the halobacterium, which were first discovered in 1940 with the type species H. volcani, the most common one used in all applications, discovered in 1975 in the Dead Sea. They exist in such high salinity environments that actually in distilled water these cells would lyse due to osmotic pressure. Within the cell, they have a light-mediated proton pump called bacteriorhodopsin, which is able to pump protons through the extracellular milieu and into the environment, whereby they can then be used in ATP synthesis via ATP synthase. The fact that bacteriorhodopsin is completely different to chlorophylls is really cool because it shows that light-mediated proton pumping activity and generation of energy via light has evolved more than once, twice as far as I'm aware, but rhodopsins are actually very common in all cell types, but these specific types of rhodopsins are only unique to archaea. Up next are my favorite type of archaea. These are Pyrococcus in the order Thermococcales. These are hyperthermophiles, anaerobic only, and they are 7 species currently known. They have a polar grouping of flagella, and as you can see in the image, they are very, very long, and they are principally peptide fermenters. Actually, in place of molybdenum, they can use tungsten in certain enzymes, formate dehydrogenase and aldehyde ferredoxin oxidoreductase. DNA polymerase has been posed as a replacement for Taq polymerase, and its proof-readability and is more thermostable. Fusion actually uses the DNA polymerase from a related species, Thermococcus littoralis, which we'll go on to in a second. Because they are thermophiles, the biotech industry is really interested in having them around. Having enzymes that work at higher temperatures can sometimes be ideal. Now we have the sister genus Thermococcus. These guys are also strict anaerobic thermophiles and they live all around the world. They have a strong sulfurous metabolism and are actually able to reduce elemental sulfur because they have an increased amount of disulfide bonds in their proteome, because this has been linked to increased thermostability of protein. You can really start to see why biotech companies would be really interested in Pyrococcus and Thermococcus because of the increased thermostability of their proteins. In fact, one of their proteases, TKSP, from Thermococcus kodakarensis, is able to actually degrade prions in the laboratory setting. Next up is the Taq superphylum. Taq comes from the names of the phyla in this group: Thalmarchaeota, Agararchaeota, Crenarchaeota, and Corarchaeota. These are a superphylum because they are more related to each other than they are to archaea outside of this grouping. Now, as of 2021, Taq is technically no longer a valid name because Thalmarchaeota has been renamed to Nitrosphaerota and Crenarchaeota has been renamed to Thermoprotea. However, you will still see the grouping called the Taq superphylum, which is why I have included it like this. Nitrosphaerota were isolated from marine sponges and originally put into the Crenarchaeota phylum, but analysis of their genome revealed a deep and persistent branching away from other Crenarchaeota and so were put into their own phylum. This phyla is perhaps one of the most abundant archaea on the planet, and genome mining has shown that most of these organisms are primarily auxotrophic aerobic ammonia oxidizers. They're capable of generating oxygen from nitrate, making them the only organism able to generate oxygen without the presence of light energy. This image on the right is a transmission electron microscopy image from a really cool paper called Spindle-Shaped Viruses Infect Marine Ammonia Oxidizing Thalmarchaea, which was in 2019. And these are actually viruses on top of the archaea, and I will be doing a video on archaeal viruses in the future, so please stay tuned for that. Corarchaeota were the first phylum outside of the Crenarchaeota Euryarchaeota duopoly that existed at the time, with the first species, Corarchaeota cryptophilium, being isolated from a Yellowstone hot spring in the Hidayan Valley in 2008. Its gene and protein conservation systems are like that of Crenarchaeota, with its core genome being 84% shared, but conserved necessary systems such as cell division, DNA replication, and tRNA maturation are more like that of Euryarchaeotas. Cryptophilium has a diameter of 0.16 to 0.18 micrometers with variable lengths. For reference, E. coli is 0.25 micrometers wide. Pseudomonas aeruginosa is 0.5 micrometers wide. Mycobacterium tuberculosis is 0.2 micrometers wide, and Mimivirus is 0.7 micrometers wide. Now is the Deepan superphylum. Like Taq, these phyla are more related to each other than they are to other archaea. Deepan stands for Diphtherites, Parvarchaeota, Enigmarchaeota, Nanohaloarchaeota, and Nanoarchaeota. For Deepan, I think by far the coolest guys are the Nanoarchaeota equitans, which are an obligate symbiote maybe to Ignococcus hospitalis, which is a type of Crenarchaeota, and it's the only member of its phylum currently known, and it was discovered with its host in an hydrothermal vent. These guys are super cool because they have no lipid or nucleotide biosynthesis ability and completely depend on their host for it. The reason why they're not considered parasites and they're considered symbiotes maybe is because they don't actually slow down the rate of growth of Ignococcus, but they are associated with stationary phase Ignococci growth. They have the smallest known genome of 480 kilobases, which is even smaller than Mycoplasma genitalium. And using rRNA, they were placed into a brand new phyla. However, some people place this genus into the Euryarchaeota kingdom in the Thermococcales phylum, because when you compare the ribosomal proteins against other known ribosomal proteins, it leads to a large number of hits against Thermococcales. Its Ignococci host is very interesting in its own regard, because they are one of the very few archaea without an outer membrane. I will be going further into the interactions between Ignococcus and Nanoarchaeota in their own video, because I think they really deserve a special spotlight. Last, but certainly not least, is the Asgard superphylum, so named because every single phyla in this group is named after a god of Asgard. Aside from Wukong, who is a Chinese monkey god from the Journey to the West, but you know what? He's welcome as well. All gods are welcome in Asgard. This is the paper that started it all. This is the paper where in 2015 they went by an underwater vent called Loki's Castle, which is just, I say just, very far north of Norway. This is where they discovered Loki Archaeota, which was very interesting because it contained various eukaryotic-like genomic features, and at that point in time was the closest known relation to eukaryotes. About 3% of proteins in its genome were that of eukaryotes, 22% bacterial, 26% archaeal, and 33% were of unknown functional homology. The eukaryotic-like proteins were involved in membrane deformation, cell shape formation processes, and phagocytosis. This diagram is the coolest one to come out of the paper. It shows about protein homologies and where they lie in comparison to other types of archaea, with Loki Archaeota at the top because they are the most closely related to eukaryotes when looking at the proteins that they share. They have an actin homologue, which is more closely related to eukaryotic actin than it is to archaeal crenactins, but also they have proteins with gelycine-like domains, which are these amino acid repeat regions that are part of the actin binding processes. For example, Sec23, Sec24 proteins have them, and they're involved in the formation of transport vesicles from the ER. But they also have vilin-gelycine superfamily of protein domains, which compose key regulators in actin filament assembly and disassembly. The existence of these proteins, later on we prove, in fact, that they have a dynamic actin cytoskeleton in Loki Archaeotum. Here we can clearly see the existence of a dynamic actin cytoskeleton in Loki Archaeota. This is not actually actin, this is what is called Loki actin, but the structure of these is very similar to the architecture of the filaments that F-actin produces. But what's very notable here is that the existence of a dynamic cytoskeleton actually predates the emergence of the first eukaryotes, and this is a crucial feature in the evolution from Asgard phylum into eukaryotes. The same group came out with this paper in 2019, the proposal of the reverse flow model for the origin of eukaryotic cell based on the comparative analysis of Asgard archaeal metabolism. And my god, is this a good paper. So what even is the reverse flow model of eukaryotic evolution? Well, the question is, when did the mitochondria join the eukaryotic cell? Because that is what makes the eukaryotic cell eukaryotic. The reverse flow model posits that H+ ions were not taken from the bacterial symbiont of the archaeal cell, but instead given to the bacterial symbiont. Loki Archaeota and Thor Archaeota are able to fix carbon. They can turn CO2 into organic molecules, but Heimdall Archaeota and Odin Archaeota cannot. Instead, they're able to use oxygen and nitrate to ferment organic molecules. Previously, it was thought that the reason why we picked up a bacterial symbiont is that we needed H+, but it seems instead that giving the symbiont H+ was able to create a beneficial energy exchange to allow it to stick around and reduce its genome. Instead, these are able to use oxygen and nitrate to ferment organic molecules such as peptides and nucleotides, and this produces hydrogen. Previously, it was thought that the reason why we picked up a bacteria was so that we needed H+, but instead it seems that we were giving the symbiont hydrogen in exchange for something else, which creates a beneficial energy exchange. So at the end of the paper, they propose again that eukaryotes come from Asgard Archaeota rather than being a separate domain of life. This is a super interesting paper, and anyone interested in the evolutionary history of eukaryotes needs to read this paper. Actually, what I would say is that it's almost essential for anyone who seeks to understand the origins of eukaryotic life to read this paper and understand what archaea are and how they came to evolve. I don't want anyone to think that this is a settled debate. This is not so. There are still people who argue for the three domains of life paradigm, and the reason for this is because they're not looking at a purely genetic level. What they're looking at instead is how these modes of life exist and whether or not we should classify them separately regardless of who evolved from who. And I'll be looking further into this question in a later video, so please stay tuned for that. Just before I sign off, I would like to just shout out some excellent papers. There are 4 reviews here, 1 article and 1 historical perspective. I recommend people to read all of these things because I think they're excellent in helping you understand the diversity of archaeal phylogeny and the history of their discovery. Thank you all for watching. Like and subscribe for Paracoccus furiosus.